My name is Mallory Williamson, and today's date is 12/18/22. My presentation is going to be on the enteric nervous system, specifically talking about short and long reflexes. What is the enteric nervous system? The enteric nervous system, shortened as ENS, was first recognized more than a century ago when scientists noticed that isolated sections of intestine removed from the body created a reflex wave of peristaltic contractions when pressure was increased on the lumen. They observed the ability of the ENS to carry out a reflex independent of control by the central nervous system, so the brain and accompanying neurons were not included in the reflex. The ENS receives stimuli and it acts on it. It controls motility, secretion, and growth of the digestive tract. There are some similarities between the enteric nervous system and the central nervous system, and in this slide I kind of listed some of those similarities, but touching more on specifics to the GI tract. So the first one is intrinsic neurons. The two nerve plexuses of the digestive tract lie completely within the wall of the gut. The second similarity are they both have neurotransmitters and neuromodulators, and among the best of the GI tract are serotonin, vasoactive intestinal peptide, and nitric oxide. Number 3 are glial support cells. Glial cells of the ENS are more similar to astroglia of the brain than they are to the Schwann cells of the peripheral nervous system. The fourth similarity is diffusion barriers. The capillaries that surround the ganglia of the enteric nervous system are not very permeable and create a diffusion barrier that is similar to the blood-brain barrier of the cerebral blood vessels. And the fifth similarity is the integrating center. Reflexes in the GI tract can be acted on without neural signals leaving the ENS. The neuron network of the ENS is its own integrating center, much like the brain and the spinal cord. So now I'm going to talk about short reflexes. Short reflexes integrate in the ENS, the enteric nervous system. So short reflexes are reflexes that originate within the ENS and are integrated there without outside input. The submucosal plexus contains sensory neurons that receive signals from the lumen of the gut. The ENS network then integrates the sensory information and then it initiates a response. The submucosal plexus controls secretion by GI epithelial cells, and the myenteric plexus neurons in the muscularis externa influence mobility. Moving on to long reflexes, these are integrated in the central nervous system. Long reflexes are integrated in the central nervous system. Some long reflexes originate outside the GI tract, but others originate in the enteric nervous system. And no matter where they originate, digestive reflexes integrated in the CNS are always called long reflexes. In long reflexes, the smooth muscle and glands in the GI tract are under autonomic control. In general, the parasympathetic division is excitatory and it enhances GI functions, leading to its nickname rest and digest. Most parasympathetic neurons in the GI tract are found in the vagus nerve. Sympathetic neurons usually inhibit GI function. So in summary, the ENS is a mesh-like system of neurons that governs the function of the GI tract. The ENS is capable of acting independently from the brain and spinal cord, but it does rely on the innervation of the vagus nerve in the prevertebral ganglia. The neurons of the ENS control the motor functions of the system, in addition to the secretion of GI enzymes. These neurons communicate through many neurotransmitters similar to the central nervous system, including acetylcholine, dopamine, and serotonin.